For decades, the clinical consensus on Alzheimer's disease has rested on a straightforward premise. The condition is driven by the slow accumulation of a toxic substance. The primary targets in this model are two specific proteins: amyloid beta, which clumps together to form plaques outside the cell, and hyperphosphorylated tau, which twists into tangles inside the neuron. But treating these proteins strictly as toxins creates a clinical problem. If Alzheimer's is simply a poisoning, removing the poison should halt the decline. Instead, advanced anti-amyloid therapies consistently yield only modest clinical slowing. That clinical shortfall points back to a basic biological reality. The healthy brain natively produces both tau and amyloid beta all the time. Categorizing these molecules merely as metabolic garbage obscures the biochemistry. By labeling them as waste, we stop ourselves from asking what their physiological job is in a healthy, functioning neuron. An answer to that question came from an unexpected source. Ari Rappoport was a computer science professor at the Hebrew University of Jerusalem. Over the last 15 years of his life, he constructed a theoretical framework of brain disease purely by reading the biomedical literature at an industrial scale. In 2025, his final theory on Alzheimer's was published posthumously. It is called the Unthrown switch. Rappoport's framework starts with a mechanical view of how we learn. Memory formation is not a continuous gradient. It is a discrete, two-stage structural process. The gate separating these two stages is not an electrical threshold or a timer. It is a physical object. Specifically, the neuron must assemble a plasma membrane's Lipid-Raft, a dense, highly ordered patch of membrane rich in cholesterol. Under this framework, Alzheimer's represents a brain mechanically arrested between two stages of memory, where the recognizable pathology is the debris of repeated failed attempts to bridge the gap. To see how this breaks down, we have to trace the two distinct stages of memory. Stage 1 is candidate generation. This is the neuron's immediate, indiscriminate structural response to a novel experience. Creating a new memory requires physical changes to the synapse, but a stable cellular structure cannot generate new geometry. To grow, the neuron has to loosen its existing architecture first. This is the actual physiological role of tau. To release the rigid actin microtubule crosslinks holding the synapse together, the cell deliberately phosphorylates the tau protein, causing it to detach. While the structure is loosened, the amyloid precursor protein, or APP, participates in this destabilized state. It undergoes what is known as alpha cleavage, sending out signals to request the metabolic resources needed for new growth. Simultaneously, calcium-permeable AMPA receptors flood the local area. This maintains a highly plastic, candidate-rich state where multiple new synaptic connections are proposed. Phosphorylated tau, therefore, is not an intrinsically pathological event. It is the required default state of any neuron actively proposing a new memory. Eventually, the brain selects a winning synaptic candidate out of those proposals. This initiates stage 2, competition resolution. The neuron must now structurally lock in that specific connection for long-term memory. Stabilizing these new receptors and scaffolds requires palmitoylation, a chemical process that anchors proteins to the membrane. But palmitoylation strictly requires the presence of a Lipid-Raft platform. This creates a critical supply chain bottleneck. Neurons do not carry the heavy machinery required to synthesize enough cholesterol to build these plasma membrane rafts on demand. Astrocytes synthesize and package cholesterol into ApoE particles. The neuron's successful uptake of this delivery throws the switch, allowing the lipid raft to finally assemble. Once the raft forms, the stage 2 cascade executes. Tau is dephosphorylated, returning to cross-link and lock down the new structure. And the amyloid precursor protein shifts its behavior, utilizing beta cleavage to produce amyloid beta. In health, this newly minted amyloid beta has a highly specific job. It acts as a targeted removal agent, pruning away all the competing synapses that didn't win the memory race. Both stages of memory formation run as a tightly engineered biological race, and the entire sequence is gated by the physical arrival of astrocyte cholesterol. Rappoport's model pinpoints the origin of sporadic Alzheimer's disease at a single point of failure, a targeted breakdown in the neuronal uptake of that astrocyte-derived cholesterol. Advancing age gradually wears down this cellular delivery mechanism. Genetic variants, specifically the ApoE4 allele, exacerbate the decay in this transport chain. The mechanical consequence is absolute. Without the cholesterol payload, the lipid-raft cannot form, and the switch to stage 2 fails to trigger. The neuron is trapped. It cannot complete consolidation, but it does not abandon the attempt either, leaving it stranded in a chronic state of candidate generation. This stalled state recontextualizes the famous pathology. Because the structure can never be locked down, tau remains chronically phosphorylated, eventually clumping into tangles inside the cell. As the neuron desperately tries to trigger resolution, it chronically secretes amyloid beta. This results in the continuous wholesale pruning of healthy synapses, accumulating as the plaques we observe on scans. Rappoport's framework identifies the cellular attempt to compensate for a broken supply chain as the actual pathology of the disease. Because Rappoport built this theory entirely outside a laboratory setting, evaluating it requires auditing his predictions against live clinical data published after his manuscript was finalized. In 2025, a study of human cerebral spinal fluid by Boris and colleagues confirmed Rappoport's exact load-bearing prediction regarding the supply chain lesion. This chart visualizes the Boris finding. Astrocytic cholesterol efflux was perfectly intact between patient groups. But neuronal uptake via ApoE4 particles was significantly impaired. The supply was fine. The delivery failed exactly as predicted. But a separate stem cell study by Lee and colleagues tested ApoE4 in young human neurons. This graph of Lee's data shows the exact opposite. In these young cells, ApoE4 astrocytes oversupplied cholesterol, actively expanding neuronal lipid rafts. Rappoport preemptively designed his framework to absorb exactly this kind of contradiction, utilizing a concept he called double-edged plasticity alongside a specific model of a compensated neuron. In his model, if rafts successfully expand due to excess cholesterol, the neuron simply overproduces amyloid without forming tangles. The theory categorizes this as a type 1 compensated state, keeping the framework intact. But this architectural choice creates a deep structural flaw. A framework predicting pathology from both reduced and expanded rafts suffers from a symmetry problem. By designing a model that accommodates every possible direction of a lipid measurement, Rappoport purchased total explanatory coverage at the direct cost of falsifiability. While the lipid-raft switch might be flawed, independent research universally confirms his core biological intuition. Cholesterol mishandling definitively drives tau pathology. An alternative mechanism, discovered by van der Kant in 2019, explains how. When neurons cannot utilize cholesterol, they convert it into a storage form called cholesterol esters. This flowchart shows the consequence of that storage. The accumulation of these cholesterol esters physically blocks the cell's waste disposal system, downregulating the cell's proteasome. This directly causes tau buildup, entirely independent of a plasticity switch. The final legacy of Rappoport's work may be its utility as a theory of capacity, rather than a theory of disease onset. Viewing memory as a supply chain operation offers a mechanistic way to model cognitive reserve and resilience. A denser synaptic space requires less new growth per memory, reducing cholesterol demand per plasticity episode, allowing the aging supply chain to sustain function for longer. The ultimate value of the Unthrown switch is methodological. It is a strict demand that neuroscience definitively map the physiological purpose of molecules like tau and amyloid before declaring war on them as toxins.